Hi everyone, for exam 3 I've chosen to illustrate and explain how chromosomes synapse during meiosis in the presence of a chromosome aberration or abnormality that occurs in chromosomes. For this, we will be looking at two different individuals. We'll be working specifically with inversions. Inversions occur when abnormalities within a chromosome happen that can potentially affect offspring. Synapsing during meiosis is when the chromosomes line up in hopes to create the most possible pairing among genes. Aberrations which occur within the synapses can cause inversion loops or compensation loops to occur. Issues might happen if crossover happens during one of these phases. During this process, viable and non-viable gametes are a result. Viable gametes occur if all the genetic material is present and there aren't any missing or replicated copies of genes. In contrast, missing certain genes or replicated copies of genes within a chromosome result in the gamete not being viable. Individual 1 will be Megan, and individual 2 will be Anna. Despite their physical similarities, Megan and Anna were both adopted but raised under the same roof. They both have decided they want to give birth to their own children that can grow up together like they have. Due to their lack of knowledge about their mothers and their fertility experiences, they are curious about their own fertilization abilities, so they have decided to do some genetic testing. Here are Megan's chromosomes. We use end-to-end flipping to determine if there are any aberrations within her chromosomes. In this case, you see here, these two homologs don't match, with the underlined segments being the reverse of one another. The specific type of inversion is known as paracentric inversion. Paracentric inversion is a chromosomal aberration that results when a portion of the chromosome is flipped and the inverted region contains the centromere. So in this case, this occurs within the boxed segment shown here. You can see the inversion loop of the genetic sequence. The inversion loop is designed for use in order to maximize pairing during meiosis so the chromosome sections line up with one another. As seen in this illustration, the top two represent a normal homolog and the bottom two represent the inversion. Crossover occurs between J and K. When considering the 4 potential chromatids which are produced from the crossover, the results are as follows. Based on the 4 gamete results generated, only 2 are feasible. The 2 underlined here are viable because all of the genetic material is obtained. The blue chromosome is an inviable gamete because it has an extra G and H and is missing an M. The yellow chromosome is also inviable gamete because it has an extra M and lacks a G and H. For Anna's chromosomes, we utilize the flipping technique again. You notice these chromosomes are the exact same, meaning there is no aberration in this sequence. Knowing that these chromosomes are the exact same, no mutations exist, meaning that during synapsis, an inversion loop won't be necessary during crossover. Here you can see all 4 resulting chromosomes are the exact same, meaning all of Anna's gametes are viable. Considering the feasible chromatids between Megan and Anna, there is a pretty significant difference among them. Anna has more viable chromatid options than Megan has, meaning Anna will be less likely to experience fertility issues because her chromatids will produce viable gametes, ultimately leading to offspring, whereas for Megan, only 50% of her chromatids have the possibility to produce viable gametes, making it more likely for her to experience fertility issues. However, luckily for them, plenty of ways for them to both become mothers exist. I hope you enjoyed this video. Thank you.